There was a paper that was published just recently in the journal Cell Reports. And what's interesting about this is that it shows this, to invoke a concept of economics, this diminishing returns when it comes to satiety and our consumption of sweet things. So the study utilized rats. I know of no evidence in humans to support or refute this. So for now, we'll assume it's relevant in humans until we learn otherwise. But they fed the rats a diet that had high levels of sugar in the water. Now, of course, that's how a lot of people get sugar, right? Any of these sugary sodas or even juice has a lot of sugar in it. And so they had the animals drinking the sugar water, and then they tested the animal's response to a load of glucose. So then gave them a bunch of glucose to eat. And they found that before they had started this high sugar diet, the high sucrose, they had a very substantial response to the glucose, and it induced a sense of satiety. In other words, they got the glucose load and they felt really full. However, with this sugar diet, with the diet that was higher in sugar, it didn't work as well. When they gave the animals this load of carbohydrate, they didn't have that same degree of satiety. That signal, that sensitivity to the carbohydrate load had been diminished. Now, they're not using the word insulin resistance there, and I'm not going to either. They didn't really test it. But I will just say they found that there was a diminished ability to sense the calories or the energy coming from that carbohydrate, and the animals would have been inclined to just continue to eat more. In my understanding and my exploration and study of insulin resistance, really two categories when it comes to understanding the causes of insulin resistance. Now, the primary insulin resistance is a term that I use very specifically. In fact, both of these terms are very specific. With primary, I consider the cause to meet that, to fall into that category, if it is based on research that has been confirmed in all three commonly used biomedical research models. Now, what do I mean by that? As a biomedical scientist, I will use and have, in fact, published papers on all of this, three models or three ways of doing experiments. At the kind of simplest, it is just growing cells in a little petri dish in a lab. So we have a little incubator that will grow cells. We'll put muscle cells on the petri dish, or liver cells, or neurons, or fat cells, and then we do experiments on those cells. Next, you can do experiments in a whole organism, like in the case of laboratory rodents. So this is mice or rats, where you can knock out genes or overexpress genes, and then you can get all of the tissue to study the fat and the heart and the brain, stuff that you just couldn't get from humans, of course, in a perfectly controlled environment in the lab. And then next is experiments done in humans. So these are the three commonly used biomedical models: cells, rodents, humans. So the primary causes of insulin resistance are those noxious stimuli that have been shown to cause insulin resistance in all three of those models. The secondary causes of insulin resistance that I'll share with you are those that maybe have been shown to cause insulin resistance in two of the three, as we will highlight one particular example of that. Or it is actually, there's evidence to support all three, but it causes the insulin resistance by taking advantage of one of the primary causes. So it's not independent of the primary cause. I hope that makes sense.